herbicide. So I'm a professor in the agronomy and horticulture and geneticist and planting breeder, mostly work on sorghum pro milling. So, ALS inhibitor herbicides act on the enzyme acetolactate synthase, which catalyzes the first reaction in the biosynthesis of the branched amino acid valine, leucine, and isoleucine. These are amino acids that are essential for the growth and development of plants. So let's talk about sorghum. Sorghum is cultivated in a global scale with over 60 million hectares worldwide, making it the fifth most widely grown crop worldwide after wheat, rice, corn, soybeans, sugarcane. The major sorghum producing regions in the world are mostly Africa, Asia, where a significant portion of the crop is used for human and animal feed. Okay? The United States is the largest producer of sorghum, followed by India, Mexico, China, and Nigeria. So here's a map showing you where the sorghum growing areas are exist. You could see most sorghum is grown in the tropics. Some of the temperate zone, that's Nebraska is in here. And you could see we grew some sorghum in the Canada region, British Columbia, and here also in Canada. We grew sorghum in Turkey, France, Spain, Morocco, and it's growing in China. Large scale of production of sorghum in China, and mostly used for animal feed. So sorghum is a versatile crop. It has so many different uses and purposes, including for food, for feed, fodder, and fuel. Comes in different types. We have grain type, we have the forage sorghum, we have the sorghum Sudan grass as a forage crop, and we have the sweet sorghum. Grain sorghum is widely used for food, animal feed, and also 30% of the sorghum produced in the US is used for the ethanol production, while forage sorghum is primarily used for animal feed. The sweet sorghum, sweet sorghum is mainly used for bioethanol production and for the production of molasses and other sweeteners. Sweet sorghum, there are more about 2 million acres of sweet sorghum grown in Brazil to supplement the sugarcane for the ethanol production. And we are trying to, farmers are trying to use it here as a biofuel source. As we know, sorghum is one of the most drought-tolerant crops in the grass family. It's more tolerant than corn, barley, and wheat. And it can thrive in arid and semi-arid regions and require about 1/3 less water than corn to produce similar energy and nutrition value as food crop or feed crop like corn. Let's talk about the herbicide resistance. Sorghum planted in middle May grows slowly for the first 2 to 3 weeks, in contrast to corn and soybean. Okay. Usually, sorghum in the United States grown mid-May to early June, but some farmers attempt to grow it end of April or early May, so they could expand the production and the growing season. So many early season weeds, including lamb's quarter, Pennsylvania, smart wheat, common sunflower, velvet, and even foxtail grow faster than sorghum, which makes sorghum less competitive to these weeds. For this reason, early sorghum is less competitive with weeds and require early weed control to be able to stay vigorous and grow well. So effective weed control for the first 30 days will give sorghum a head start on weed, mostly grasses and dicots, and hopefully will benefit the farmers by increasing the yield and the growth. So when farmers in that state ask what is the main constraint to growing sorghum, the first one is weed control. Weed control is a major problem, especially grasses. So let's talk about weed management in sorghum. We will have pre-option, pre-emergence option are exist, such as atrazine, but sometimes in dry areas, that's not as effective because we need moisture for these herbicides to work well. Post control of for protein, we have this commercial herbicide like Dual, Magnum, P, Permint, and atrazine that are exist to control broadweed. You could mix atrazine with 2,4-D or Dicamba to control broadweeds after sorghum, about 18 inches, or what we call about 30 or 40 centimeters tall, so you could use this herbicide. So the biggest problem for sorghum that limit the sorghum production and reduce the yield is the control of grass post-emergence. So weed control, this is the objective of this study, is to develop grains, wheat, sorghum, and forage sorghum, hybrid and hybrid line that are tolerant to all classes of ALS available commercial herbicide. There are 5 classes of ALS herbicide that comprise about 60 or 65 different commercial herbicides that we used in soybean and corn and other crops. So we have identified 4 different mutants that were found in Shattercane. Our objective is to take these mutants and stack them or pyramid them together. So we have, instead of 1 resistance, you will have 4 or 3. And the idea behind it is to extend the resistance of these genes to the herbicide. What is Shattercane? Shattercane is here, like you see on the right. It's Shattercane because the seed shatter will start maturing, so just for ground compared to the grain sorghum, like you see here, standard. So Shattercane is a subspecies of the Sorghumbicolor, and it's crossed widely with sorghum. So it's good and bad. It's bad because all these shattering genes will move to grain sorghum, and it's good if we have identified any good genes in Shattercane as a forage characteristics or tolerance or resistance gene. We could easily hybridize them to sorghum and then select for the desired trait. As I mentioned, the herbicide, the ALS herbicide commercially available, these are the sample of them. You could see in here, these are the different 5 families in the ALS commercial herbicide, and this is the abbreviation of this. I'm not going to attempt to butcher this pronunciation, but I could see the SUIMA, IMI, TPS, POPs, and the SCTS. These are the different family, and you can see each family, there are the commercial herbicide that belong to this family, and in bacon, accent, pig, alley, vermin, you can see this in here, and then these are the herbicide in this family. And there are more These are the most known herbicide, these 5 families. This is a field of sorghum where you could see the weeds overtaking the crop. And it's a major constraint, as I mentioned, to growing sorghum. And the losses could go up to 97, 95, 97%, or almost a total loss. Farmers don't even harvest the field when it's like this one. And it's amount to 5.7 billion kilograms, which the value of about $953 million, or almost $1 billion. This is based on 2020 statistics. So you can see that it's if you have grasses in your sorghum, it's goodbye to the crop. So let's talk about the history of these mutants. 1992, we have collected Shattercane plants and seed from 16 different fields of soybean and corn over 3 years, and these fields were treated with pecan and accent for the 3 years. And in 1992, we screened 52,000 plants with different herbicide to see if they have any identified mutants. 1993-94, we screened additional 17,000 plants and identified one mutant. From the 52,000 plants, we identified 3 different mutants that are show resistant to different commercial herbicides. So Shattercane seed, let me see if I have a picture of the Shattercane. I did. Okay, so Shattercane seeds were collected from individual plants having survived that season herbicide application. 20 seeds from each one of these were saved to increase the seed to make it homozygous. 500 seeds from each plant were evaluated in the greenhouse condition and treated with the pyrimethamine herbicide at 1/4x, 1x, and 2x, and plants surviving the 1x treatment and 2x treatment biotype was used to screen for a final screening and to be identified as 100% mutants. So the seed from these few seeds from each different mutants, positive mutants, we have gave them name B205, B30, B910. We grown these in the greenhouse to give rise to plants, and then these plants were used to extract DNA, and the DNA were quantified, and then we used the ALS from corn, because corn is very close to sorghum, designed and synthesized 6 oligonucleotide to amplify the ALS gene from sorghum. And after we amplified with these PCR fragments, we sequence the total gene, which is about 2100 nucleotide. And then in addition to the mutants, we also identified or amplified DNA from a wild type, from commercial sorghum CK60 and from maize, and then we align these together to identify any SNPs or substitution of the nucleotide. And you can see here we have identified 1, 2, 3, 4, 5, 6, 7, 8 different substitution of the ALS gene, and these 3 different plants or mutants And these are the changes from amino acid proline to leucine, all these you can see here. And these resulted in these plants becoming resistant to these listed commercial herbicide. Okay, so you could see that P8-30 has the most resistant, it has 3 different changes between, and show the resistance to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 different commercial herbicides. So this is an example of the treatment to identify the susceptibility and resistance and tolerance to these plants. This is the treated control. These are the 205, 54, 102. This is untreated control. You can see the action of the this herbicide 2 days, 20 days after. And we have another herbicide in here, Olympus, and you could see this is treated control, this is untreated control, and this is the reaction of these different mutants. Then we have this. Let's go to. Come on. Oh, so we have a problem with that. Okay, and this one here is for different another herbicide from ALS velocity, and this is the treated control, and this is untreated control, and you can see the reaction of these different methods to this herbicide. So, as I said, we have 3 different mutants based on the sequence, and we found another mutant, so we have total 4, and now we are in the process of stacking or pyramiding these genes together, and the whole idea is to extend the resistance in these plants for a longer time, because we know that we have only 1 gene. The breakdown of the resistance is within 5 to 8 years. If you have multiple genes, you are hoping to extend the resistance to the herbicide or to the disease or to the insect if you are. So that's the idea behind pyramiding, is to extend and expand the resistance, prolong the life of the resistance. So in conclusion, with the development of ALS resistant grain sorghum, several post-emergence ALS inhibiting herbicide can be used to control weed in grain sorghum, mostly grasses like foxtail. The ALS elite sequence and DNA marker we described here in the study could be used for marker assisted selection to rapidly identify the resistance or tolerance in the hybrid and hybrid line. The significance of the study will be based on exploring the 4 different ALS mutants, which may provide an expanded the resistance, and hopefully the ultimate goal is farmer will be more acceptance to grow sorghum, and that at the end would be more profit and expand the sorghum to additional region that will grow in. Because as I mentioned earlier, the first constraint by farmers to grow sorghum is herbicide resistant. So if we could provide herbicide resistance option to the farmers, hopefully there will be more acceptance to growing. All right, so most of the work I presented to you here were published in 2023 in Hereditas. And so if you need more additional information on the study, you can find it in here. And with that, I will thank you for listening. And hopefully, if you have any question, either you could send it to me or we'll see how that will work and be happy to answer. Thank you.
Thank you all for your participation and cooperation for the success of the virtual session. We are looking forward